take it away.
So, all about Mo.
MOGAT.
Oh, Mo means multi-omics. Okay. Good morning, everybody. Okay. So this work is disclaimer. This work is done by this student. So he's the expert. He's not here. Okay. I am just representing him. Okay, as his professor. Okay. So it's an improved multi-omics integration net framework using graph attention network. So it's well known that integrating multi-omics helps in deciphering insights of complex diseases like Alzheimer's, cancers, etc. Using genomics, epigenomics, transcriptomics, or proteomics. And traditional machine learning approach cannot take advantage of this multi-omics integration because of high dimensionality and low size sample, and they cannot find or decipher the complex relationship between two omics. And recently, lot of researchers are using graph based learning approaches or graph neural network, and recently there is some work done on multi-omics integration using graph neural networks. They mostly use graph convolutional neural network. So they consider neighboring nodes. They give equal weights to the neighboring nodes. Okay, they did not take care of any attention of neighboring road. If some neighbor is more important or less important, graph convolutional neural network cannot take consideration of that. So what we are suggesting that if we can take attention of neighboring nodes, maybe it will provide better insights or better subtype classification and so on. That's why we proposed GAT based approach for this work. And another disclaimer is this is a very preliminary work. Okay, so we are just presenting. We have some improvement and we submit it to CCB. Okay, you can present it. That's why I am here. But a lot more work to be done actually. Okay. So this is our proposed approach: multi-omics integration using graph attention network. We used 8 different types of data. None of the previous study incorporate long non-coding RNA in their studies. So we are the first. Our study is the first study that include long non-coding RNA also. Okay. This is the framework. So in the first block, we have data matrix that's coming from 8 different types of data: clinical data, copy number alteration, co-expression, microRNA expression, long non-coding RNA expression, DNA methylation, microRNA, and single nucleotide mutation. These are data matrix. From this data matrix, we generate a similarity matrix. From that similarity matrix, we find 8 different networks. We call it patient similarity network. Here each node represents a patient and age represents the relation between 2 patients. So after generating 8 graphs, we use a graph attention network on top of each network for embedding, and that each GAT provides some embedding, and finally we use this embedding for downstream analysis like subtype cancer prediction, survival analysis, etc. So to develop and investigate our proposed method, we used breast cancer data for Cancer Genome Atlas, and we downloaded RNA-seq data, mRNA, microRNA, long non-coding RNA, DNA methylation, copy number variation, simple nucleotide variation, and clinical data. And this data set has about 1100 samples, but only 920 are common among those data set, and that's what we used for analysis. And this table shows the distribution of breast cancer subtype data. So, for this process, for this method, pre-processing is the most important task actually. Okay, you see this slide shows all the pre-processing steps. I am not going to discuss all of them. So, this one is for clinical data, this one is for copy number alteration, this one is for co-expression, we use WGCNA, and this is for mRNA expression, lncRNA, and so on. So, this is the final feature set. We started with original features like these numbers. Then second row represents the features after cleaning. We use some threshold because microRNA, lncRNA, they have a lot of missing values. So, we need to clean those stuff. So, after cleaning, the second row shows the cleaned features, and then we used feature selection approaches called Boruta, and last row shows the final number of features that are used for the downstream analysis. So, once we have that features, now we need to create the network. So, how do you create the network? As I told you, each node represents a sample or patient, and age represents a degree of similarity based on some threshold. So, to find the similarity, we used different kind of similarity metric because all data set you cannot use same similarity metric for all data type. Like for expression data, we used Pearson correlation. For mutation data, we used Jacquard similarity, and for the clinical data that has binary and age, so we used Gore metric. Okay. And for generating similarity patient similarity network, we use top 3 ages. Okay. That's pretty standard actually. If you use top 3 ages, it covers most relevant information. And this table shows the summary of a network topology. As you know, each network has 920 edges, and the last row represents the number of edges after taking top 3 edges for each node. So, once we have the graph, 8 different graphs, then we use GAT for embedding to extract the salient features from each of the 8 graphs. Okay. Intuitively, GAT, this is a node of interest, here the patient or sample. This is the initial representation, this is the updated representation. How we update? To update, we use every node has its features. We take care of that features, and we get information from neighboring nodes also. In this figure, we have 3 attention head. You can see. You can use any number of head attention head. So, it's collecting information from neighboring nodes through 3 attention head. And after collecting, we can do concatenation or average. Usually for intermediate layers, we do concatenation, and for the final layer, we do average. So, this is the sub comparison with our results with the existing state of the art results. MOGAT and Supreme, these are the two state of the art to integrate multi-omics data, and this is our MOGAT. As if, so this comparison using 3 data sets, and this one is using 8 data types. The reason is that MOGAT, we had some issue with the MOGAT. Actually, MOGAT they developed it for 3 omics. Okay, when we add more omics, it was giving trouble, crash. So that's why this comparison is only with 3 omics data. Okay, so our MOGAT performs better than both MOGAT and Supreme. But for Supreme, we are able to use 8 data types, and in case of 8 data types, our MOGAT performs better. Okay. So as I said, this is very preliminary data. We need to do a lot of down the stream verification. Okay. So next question is, you are using so many data types. Do you really need all the data type? So this table shows that yes, we do need all the data types. It gives some kind of improvement. If you see the last row, the performance using all data types, and other rows are performance without one data type. Like these are with 7 data types. Like first row, all except clinical data, and then second row, all except copy number alteration. You see all these performance are less than the 8 type of data types. Okay, that tells yes, they all data types play some role. Okay, and these two are highlighted. You see this one, if we exclude mRNA expression data, you have the most drops from the combined results. What does it mean? That we can tell that mRNA expression provide the most information. That's what I can tell. And microRNA provides the least information towards the integration. So then we did survival analysis. This is the one validation of our work using raw and our embedded features. So we use lasso, Cox proportional hazard ratio, and then Kaplan-Meier plot. As you can see, using raw features, this survival analysis, it does not produce good results compared to our embedding. Our embedding can differentiate high L, low risk groups with high significant and high hazard ratio. Hazard ratio 0.1, that means it's pretty good separation you can see between high group and low group. Okay. So in conclusion, our proposed approach MOGAT performs better than the state-of-the-art multi-omics integration in cancer subtype classification and survival analysis. And we have not tried this model for other cancers. That is our next steps. I'm sure it will perform good on other cancers or other disease type also. Okay, that is our future work. So this is my group, machine learning and data analytics groups. These are my students, 3 students, PhD students. They are working with me, and this is my collaborator in this paper. He is also from our department, and we are also part of a big group, bioinformatics research group under a professor, and this work was supported by NSF Career Award. Okay. Thank you, everybody.
very much for the talk and for making it.
Okay.
And we got time for a couple of questions.
Yes, please.
Oh, I have a quick question on the hyperparameter optimization and the comparison. So I'm part of this collaboration that's trying to compare different, multi-omics integration methods, and we found that not all of the tools are necessarily hyperparameter optimized. So the comparisons are not, it's hard to know, right? Because they're not all optimized sort of equally. So did you optimize all the tools that you were comparing to, and how did you do that?
MOGAT, we did optimize for our one, and Supreme, yes, we also did Supreme optimization, but we could not do optimize the MOGONAT because of some technical issue. Yes, you're absolutely right that when you compare these deep learning things, every model has to be optimized. You're absolutely right. Yes. Thank you. Yes.
Yeah, you go.
So, in the survival analysis, you compared embedded features with the
Raw features.
But the embedded features, when they are created, they take the clinical data as well as input, right? And the clinical data actually involves the survival data. So, did you exclude that when creating the
Clinical data, we do not have survival component.
Okay.
Julio, you?
Can you go back to the example where there was this performance against the other methods back? So here, like on the left is one with three.
3 omics.
Yeah, on the left, also on the
Right.
right with many more.
8 omics. Yeah.
And does it look very different, the performance on the model working with 8 omics instead of 3?
It's slightly better.
And so my question is, like, do you think it's worth to put the money to get all those other omics to make that?
Yeah, that's a good question. Whether we should take consideration of all the omics data. Even though it contributes, yes, it's a dilemma actually. It contributes little bit, you see, and the 7 omics, it's very close to this. So we might find your question is probably maybe if you use 4 omics, we might get, I'm not sure whether we'll get better than this. I'm not sure. We have to do that kind of experiment actually. Okay.
All of these also got error bars that are bigger than the difference.
Yeah.
Question
Did
there?
did you also look at subtype specific F1 in your comparison?
Oh no, that's a good question. That is part of our future. You're talking about this. Am I right?
So you looked at the overall
Overall,
comparison.
yeah.
Specific
Yeah.
performance
No,
comparison.
we didn't do that. Yes, that's my student is doing. That is my next. Yeah.
One question there at the front again.
Yes.
Just a little question. I saw you used the multiple omics data. Have you tried also to consider gene dependency? Or
Gene dependency. So you're talking about the individual gene.
yeah, I mean, I saw the extraction, you used multiple stuff like that. Also gene dependency.
What exactly you mean by gene dependency?
Is about the dependency of that gene in the cells. How is good for the survival of the cell.
So I think the question you're asking that goes to the feature selection. Like we have 20,000 genes, use some feature selection approach, reduce to maybe 100 significant genes, then you do the survival analysis.
Yes.
That's what you're talking about. No, it's not here in this study actually. We do those kind of things, like I have some other work with just feature selection. So we published in International Journal of Molecular Science. Yeah, that one we reduced the genes from 20,000 to I believe it was around 60s. Then we do the survival analysis. So that is what you mean by gene dependent probably. Am I right? Yeah. No, this one is embedding. After embedding, we do not know which genes are there actually. Okay. Embedding, it just gives us salient features. Okay.
Okay, so if there are no more pressing questions, there are pressing coffees, I guess.
Yes. Can we only ask the speakers of the next session to come up for a second here, and so we can check the slides before?
So let's thank all the speakers of this session.